So as I'm sure you're all also aware, synapses are lost in the Alzheimer's disease brain. We've known this for a while, and this happens as an early event in the disease process. And synapse density is one of the greatest correlates to cognitive function in Alzheimer's patients. Now, unfortunately, it's not quite as simple as just synapse loss. So when I was at Lilly, we wrote a review a few years ago where we described the different types of synaptic plasticity, and then we described what actually happens to these different types of plasticity in Alzheimer's disease. And you can see here that there is the structural plasticity, so this is where the density of synapses, and we know that they are lost. This can also include stability of synapses as well as their molecular composition. We then move through to synaptic plasticity, which is how the synapses function, either at the single synapse level or in pathways in the brain, before moving on to the connective plasticity, and this is then the activity of the whole network, before we then look at the behavior of the individual. Now, all of these processes are affected in Alzheimer's disease and other types of dementia, and they are affected by a number of different mechanisms. And there is a complex interplay between intrinsic and extrinsic mechanisms. And I've just listed a few here. This is by no means an exhaustive list, so everyone will find something I've missed, I'm sure. But you can get intrinsic mechanisms. So this is the health of the neuron to which the synapses belong. Breakdown in transport, proteostasis, so that the synapses do not have the proteins they need to function. And this then occurs alongside extrinsic mechanisms, such as glia, not only microglial pruning of synapses, but also the support that many types of glia, particularly astrocytes, provide at the tripartite synapse. And then we need to think of synapses not in isolation, but at the network level, where there is a delicate balance of excitatory and inhibitory neurotransmission, and this balance is affected differentially at different stages of the disease. And then bear in mind that synapses are inherently plastic, and this is crucial for learning and memory processes. And then underpinning all this is the pathology. And we know that synapses are affected by pathology as it accumulates as the disease progresses, but also they are affected when they're in close proximity to pathology, such as amyloid plaques. And then nice work coming out from the Tara Spires-Jones lab looks at then hyperphosphorylated tau or beta amyloid directly at synapses, and understanding the effect there at individual synapses will be key. But we need to take a step back. So all these things go wrong, and different types of plasticity, synaptic plasticity, are affected. So I think we need a consensus in the field about what we are actually trying to achieve when targeting synapses. Again, in our review, we discussed that you do lose some synapses during healthy aging, and we know if Alzheimer's is left untreated, there is a significant loss of synapses. We hope that with disease modification, we can ameliorate the synapse loss, but what we want to do is develop therapies that we can actually either initiate a plasticity response or a neuroprotective response to boost the synapse numbers and function up towards the healthy aging level. And there have been ways to do this in the past, and that is to increase or enhance the synaptic efficiency. But we are thinking more longer term, more neuronal remodeling, how we can either boost the numbers of synapses, boost the certain types of synapses, or their molecular composition, as well as providing the necessary support they need to function correctly. And to do this, I think we need three things. One is a pharmacodynamic measure. I think we need to begin to reach a consensus in the field about what it is that we actually want to be changing in disease. We also need to know that it is whether that pharmacodynamic measure is actually critical for cognition. A lot of my work is synaptic proteomics, and we have some evidence that some change and some proteins change, and that may not actually affect the cognitive ability. And then lastly, we need a reliable biomarker, and I will touch upon this in a second. So we have addressed this in a number of different ways. I'm just going to show one or two quick bits of data. This is where we've isolated synaptoneurosomes from late-stage Alzheimer's patients, and we've looked at the proteomic composition in different brain regions. And we found that even in the visual cortex on the left-hand side, which is affected relatively late in disease, we still see synaptic proteomic changes. And then we see these in the other regions, which are also affected at different stages. And we can then look at the pathways that these proteins belong to give us an idea, a pseudotemporal profile of what the synaptic proteomic changes are. We also can look at different types of synapses, and synaptic proteins rather, and you can see here, unfortunately I don't have a pointer, but on the graph on the right, that in the prefrontal cortex, a region affected at mid-stage disease, this is where most of the inhibitory synaptic proteins are affected. Now, this was a pseudotemporal profile, so we wanted to check whether this was actually occurring in earlier disease, and we looked at BRCA3/4 stage tissue, and again we found that mitochondrial pathways, similar to what we were seeing in the visual cortex, were apparent there. We then want to know whether some of these signatures are crucial for cognition. So a bioinformatician in my group looked at bulk protein and transcript data and derived a synaptic signature and then mapped that onto the ROSMAP cohorts, which are very well phenotyped. And there she found that in the ROSMAP cohort, there's actually 2 rates of disease progression, and that when she mapped the synaptic module onto those, you can see that there is a difference between the slow and the fast progressing patients. Now, obviously, what would have been nice to do in this study is to have a biomarker. And there are a number of different either fluid biomarkers or PET biomarkers now coming out for synapse density. We've been investigating SV2A, which is the target of a PET biomarker, PET ligand. And here, my placement student stained synaptophysin with SV2A to see whether SV2A was ubiquitously expressed at synapses. And he found that actually the proportion of SV2A co-localizing with other synaptic proteins, in this case synaptophysin, was actually pretty low, around the 20% level, a lot lower than we expected. He then found that SV2A has a greater propensity to co-localize with excitatory synapses. And then we asked, well, if it's not always at synapses, where is SV2A? And we found, to our surprise, there is a considerable proportion of SV2A in astrocytes, and this actually increases in Alzheimer's. So just starting to wrap up, when Jeff Cummings does his annual review of clinical trials, about 12% are targeting synaptic targets. And this is, I analyzed these over the last 5 years, and this is pretty consistent, maybe with a slight dip. And these actually target, there's a range of different targets, and they include some repurposed drugs as well. So this work is ongoing, but we aim to improve the outcome of these trials. And to do that, as I said, I think we need these 3 key things, but we also need to bear in mind lifestyle factors, and we think that these will be used as combination therapies. And I want to stress that as much as possible, this should all be human-led and reverse translation either into animal models or into iPSC cells, for example. And then I think we can move forward with developing synaptotherapeutics. And thank you for your time and attention.
We'll now move on to our second speaker, Professor Adam Greenstein from the University of Manchester, who's going to challenge the neuronal focused model and bring in the vascular dementia and Alzheimer's disease. Adam.
Okay. Timing. Okay, thanks very much. So my name's Adam Greenstein. I'm also a consultant geriatrician and a high blood pressure doctor. So when you combine those two specialties together, you get brain illnesses. So I was just trying to calculate, and I think probably over the last 20 years, I've told probably about 500 people that they've got dementia and that there's no treatment. So I'm representing the group in Manchester. We do vascular brain health research. I sit at the center of this ecosystem. From a scientific perspective, I do vascular biophysics and calcium imaging of small arteries. I'm just going to start with a couple of potential clinical scenarios, though. Imagine if you went to a doctor with central chest pain and some breathlessness, and they looked at you kindly and said, "Oh, I'm not sure that there's much wrong with you, but come back after you've had your first heart attack, because it's not really a disease of the arteries, it's a disease of the heart tissue, and we don't know how it occurs." Let's move on to the brain now. Imagine if you went to a doctor and you said, "About a week ago, my right arm was weak, I had slurred speech and difficulty understanding words." And they looked at you and they said, "Well, come back after you've had your first stroke, because it's not a disease of the arteries, it's a disease of the brain. And we don't really understand how it works." I've done one for renal, but I'll just move through that. The thing about vascular illnesses is this. You've got to start early, you have to start aggressively, and you've got to give people a multimodal approach involving blood pressure, cholesterol, diet, obesity, alcohol, smoking, and exercise. If you don't do this, you get irreparable damage to the target organ. So let me give you an example of another vascular illness. So you can see that memory loss and dementia, all of your risk factors are vascular. And the ones that don't look like they're vascular on that list are actually your sensors. And it's your sensors which stimulate blood flow to the certain areas of your brain. We call this neurovascular coupling. So every single one of those risk factors is vascular for dementia. But we don't do anything when people present and tell us that they've got something wrong with their brain. We tell them to come back in 5 years' time, and then we tell them that there's nothing that can be done for them. And that treatment is palliative because we don't know what's gone wrong with the brain. So from our perspective in the lab, when we study arteries of the brain, we think that the brain looks like this. Each of you have got more than 1,000 miles of blood vessels in your brain when you include the capillaries, and in fact you've got more vascular cells than nerve cells. And when you look at patients who present with memory loss, whether this is Alzheimer's or vascular, although what I'm going to tell you is that there's very little distinction between the two, is that you have a reduction in brain blood flow which precedes the diagnosis of dementia. An indicator of more severe disease and more amyloid, and you've got impaired neurovascular coupling with a reduction in brain blood flow. So let's just talk about one of our clinical studies, a SPRINT study. This came out about 6 years ago. Showed that if you reduce blood pressure, you reduce the onset of mild cognitive impairment, just mild cognitive impairment, over 3.5 years by 20%. Now this is very important. The people in that study had their blood pressure reduced from 133, which is probably, I would say, maybe about a third of you in this room, to 122. That's the biggest effect on brain health for any clinical trial ever. So that's the SPRINT Mind study, and just for the sake of clarity, these are patients who are over 65, they're 67 years old. Here's another study from the Lancet, the finger study. This one's a multidisciplinary study focusing on cardiovascular disease. And when they did that, including lifestyle, exercise, and diet, again, you get a 20% reduction in functional disorders. And if you look at the longitudinal studies, like the Whitehall study, it's extremely clear. In the over 65s, when your blood pressure in midlife is above 130, that's when you start to develop small vessel disease of the brain. And just look at what happens when you put all of these studies together in a meta-analysis. The lower the blood pressure is across the clinical trial, the less the development of white matter hyperintensities you get. So as a cardiovascular physician who treats high blood pressure and treats elderly people, you can see that the diagnosis and this concept that somebody might have Alzheimer's disease or vascular dementia, depending on what an old age psychiatrist thinks that they might have sitting in a room in a small cubby hole in a community hospital, is complete nonsense. And in fact, when you do post-mortem studies, they're wrong 50% of the time. In my patients, over the age of 65, all of them have got small vessel disease of the brain, and about 40% of them also express amyloid beta. This leads to small vessel disease of the brain, which reduces brain blood flow and causes memory loss. And this is about 80% of the population over the age of 65. So, like I said, after about 20 years, you get really tired of telling people that they've got a terminal diagnosis when you know you should have been seeing them about 5 years earlier, when, like with all vascular illnesses, you should have intervened at that point, because let's be honest, the evidence is there. So in Manchester, we've set up the very first brain health centre where we see people with mild cognitive impairment and we implement a holistic cardiovascular intervention program. We do this in tandem with the old age psychiatrists, and we do this because they have a reduction in brain blood flow, they have a reduction in functional hyperemia, they've got an excess of cardiovascular risk factors, and believe it or not, of all of the patients who have a diagnosis of dementia, only about 0.5 of them, so 1 in 200, get a tissue diagnosis of whether they have dementia or not. This is the first clinic of its kind anywhere in the world, and we're measuring brain blood flow using MR scan. So we call this Brain Health Manchester. It's based upon these 3 pillars here: early molecular and imaging diagnosis. So we're actually diagnosing the disease that people have, and access to, as Joanna was saying, the new disease modifiers. Because the reason that NICE gave for not actually allowing people to use these drugs is because there's no infrastructure to give them. We address treatable cardiovascular risk factors, and we have seamless access to research. Like I said to you, about half of my work is involved in the ion channel biophysics of small arteries, how calcium signals connect with potassium channels in order to regulate contractility of small arteries and facilitate neurovascular coupling in the brain. And this is our patient pathway. We're doing it in conjunction with one of the UK's largest brain health charities, and within the confines of the Geoffrey Jefferson Brain Research Centre in Manchester, which I'm sure some of you will have heard of. Patients come in the morning with their families, they get their scan, or they've had their scan a couple of weeks before, they get a cognitive profile. Myself or one of my staff will do an advanced hypertension clinic, and then they have a lumbar puncture so that we can actually tell what they have. We do the advanced diagnostics. We set up the second lab in the UK now, where we can measure amyloid 1 to 40, neurofilament light, phosphorylated tau in their CSF, which we're storing for subsequent studies. And so I'm going to just go back to what happens if you don't treat vascular disease early. Catastrophe. And that's what happens, because that's where we are right now, unfortunately, for all of the beautiful research that's going on in the preclinical arena. patients as just coming in and being told that there's nothing they can do because we're diagnosing them too late. Thanks.
Eventually. Now, so we've now got Will McEwen from Cambridge, who's going to give a more immunological perspective. Thank you so much.
Thank you, and thank you for this opportunity to present. So I'm Will McEwen at the UK Dementia Research Institute at the University of Cambridge, and we're working on innate immune control of protein aggregation, particularly tauopathies and Alzheimer's disease. So tau, as I'm sure you're all aware, is abundant pathology in Alzheimer's disease and a group of about 20 other diseases known as the tauopathies. It forms these abundant aggregates inside neurons in Alzheimer's disease, and at the molecular level these are these straight or paired helical filaments. And more recently the atomic resolution structure of these filaments has been resolved, demonstrating these layered beta sheet filaments stacked on top of each other, forming these very long elongated filamentous structures. And as Joe was saying earlier, this correlates very well with pathology. So the amount of tau you have on the x-axis there correlates very well with the cognition at death in Alzheimer's patients. So in terms of how tau aggregates, so we know it as a native protein, it's structurally disordered. It undergoes some kind of oligomerization to form small assemblies. And some of these assemblies have a conformation that are compatible with elongation, so monomer addition to form elongated fibrils. If these fibrils then fragment, you get a doubling of the number of growing ends and the potential for exponential amplification of the aggregated mass. And as this occurs inside neurons, if one of these aggregates can escape from one neuron and get inside another one, it can then template that aggregation elsewhere. And this kind of aggregation is consistent with the way that tau is known to spread around the brain during Alzheimer's disease, and is also supported by these animal model experiments where injection of tau filaments into animal brains causes aggregation to occur. So in recent years, we now know that targeting extracellular aggregates is possible. So lecanemab and donanemab, for instance, have reduced beta amyloid very effectively in the extracellular space. But for the proteins like tau that are in the intracellular space, and this includes TDP43, alpha-synuclein, and various others, there really remains very few ways by which they may be addressed. So what my lab's been doing is asking whether we can take antiviral pathways, which are present inside our cells, and redirect these pathways to degrade protein aggregates. And the gene we've been mostly interested in is a gene called TRIM21, which encodes an intracellular antibody receptor. And this is a very unusual location for an antibody receptor because antibodies aren't normally inside cells. It's also unusual in that it has E3 ubiquitin ligase activity. So at its C-terminus, it has a pri-spri domain, which is a protein fold usually that mediates protein-protein interactions. And this pri-spri domain can interact with the antibody FC region. And when it does so, it can activate and mediate the destruction of virus antibody complexes. So in a series of papers over the last 15 years or so, we showed in Leo James's lab at the LMB that viruses can import antibodies with them during infection. TRIM21 will then bind onto those antibodies in the intracellular domain and destroy the virus antibody complex at the proteasome. So we got interested in whether this process may have a role in tau therapy based on these papers that showed that various ways of immunizing animals against tau pathology could reduce tau pathology in vivo, these preclinical animal models. So an experiment that we did was to ask whether, like viruses, tau assemblies could import antibodies with them into the cells. So these are neurons incubated with tau assemblies, which themselves have antibodies coated onto them. As you can see here, these tau assemblies make it inside the cell with this antibody, in this case called BL134, inside the neuron, and TRIM21 latches onto it. In vivo, we showed that when we when we passively transferred antibodies into these animals, we were able to protect against tau pathology. So on the left there, in the TRIM21 animals, animals that contain TRIM21, but in animals that lack TRIM21, these antibodies were no longer protective. So immunotherapy in mice requires TRIM21 in order to be effective. So we're now taking these ideas forward, really telling us that we can do something, and also gives us a mechanism by which we can start to optimize antibodies to engage this mechanism better. Secondly, we've taken this forward into a kind of more targeted protein degradation mechanism. And we made this observation that TRIM21 on its own is inactive as an E3 ligase. It's only when it engages a multimeric complex bound by multiple antibodies that the ring domains become engaged with each other, cluster, and become activated. So this gives us a mechanism by which we can start to try and encode aggregate selectivity. So we took the essential components of TRIM21, which is the ring domain, its E3 ligase activity, and combined it with the essential activity of the antibody, which is just binding. So we made a ring nanobody fusion. And the idea is that this would only become active when it bound to an aggregate. When it binds to monomeric tau, it shouldn't be activated based on this clustering activation. We can also target the tau aggregates by fusing the TRIM21 ring domain to tau itself. And this again should only become activated in the context of an aggregate. So this is a field of cells expressing aggregated tau, and at the start of the video, we started, we induced expression of the degrader. This is a ring nanobody degrader. And over a period of a few hours, we saw the tau aggregates being removed very effectively. Quantified on the right there. If we did the same experiment in cells expressing soluble tau venus, however, we saw no degradation of the antigen, confirming that we were able to get this aggregate selective degradation inside cells. We worked with David Kleineman to do some single particle super resolution imaging, and found that when we have expression of these degraders, we saw degradation across a range of sizes, but particularly the longer ones were degraded. So what we have, therefore, is a very unusual type of degrader, where the larger the target is, the better the degradation. We were able to package our degraders inside AAVs and inject them into mice. These AAVs have the ability to cross the blood-brain barrier. You see at the bottom there we have very good global coverage of the CNS. And at the top, sorry, I don't have a pointer, but on the right two are the control conditions, and on the left is the one that's been treated with the active degrader. And you can see that the amount of 88 positive tau pathology is substantially reduced. This was also had effects on the motor function in mice. So these P3ONS mice that we are using at 4 months can walk across a perspex bridge like this, but at 6 months have a locomotion defect and take more steps and longer to cross the same walkway. However, a treated mouse, now treated with a degrader, is able to cross similar to the 4-month condition. And that's quantified here. So the deterioration that the untreated mice get is prevented in mice treated with the active degrader. So to conclude, we've shown that there's an intracellular pathway. These antiviral intracellular pathways can be used to remove tau aggregates from the brain. And first, in immunotherapy, we found that TRIM21, in its natural role as an antibody receptor, can be used to degrade these intracellular immune complexes. And this gives us a new way of potentially optimizing antibodies using the TRIM21 pathway for therapeutic potential. And secondly, as a targeted degradation approach, we've shown that we can engineer ring nanobody and ring bait degraders, which when incorporated or bound to tau filaments, elicits their selective removal and improvements in animal behavior. And this gives us hope that we may be able to use this type of pathway for reversing of this kind of pathology in vivo. So I would like to thank all my group, and thank you very much for your attention.
Thank you, Will. So we're now going to hopefully invite, well, not hopefully, we're definitely going to invite the speakers back up to the stage and take questions. We've got, I think, you guys can have this in a second, but I think there's another mic as well that we can pass between you. And also, there's going to be roving mics coming around. And yeah, so basically, if you've got any questions or, you know, controversial points you'd like to put to the panel or to points you'd like to make yourself, then to discuss where we might get the most promising breakthroughs to treat neurodegeneration, that'd be great. So I'm going to hand over to, well, any of you, if we've got any questions. We seem to have a question right at the front. Hang on. Oh, you're both internal. So.
So I was really interested, know nothing about neurodegenerative diseases apart from what you read in the news. And I was really interested, A, that you said about the vascular, and B, that you talked about lifestyle. So if you look at lifestyle, we see a huge increase in type 2 diabetes and obesity, and I believe with type 2 diabetes you have vascular issues as well. So with the introduction of GLP-1 to treat obesity potentially, do you think that tackling some of these lifestyle issues like obesity, like type 2 diabetes, will improve vascular health? So do you think these drugs like GLP-1 will be seen to improve vascular dementia?
Hello? Okay. Yeah, thanks for that. That's a really wonderful point, and it's a very exciting time, actually. I think I'll just make two very quick points about the GLP-1 agonists. And I think the first one is that increasingly across the medical and scientific spectrum, we're seeing that these drugs don't actually target obesity per se. They more target addictive behaviors. So we're using them for all sorts of things, and this will be kind of forthcoming. You can see that people who take them, they drink less coffee, they smoke less, they stop smoking, they stop eating. I don't think it's about eating per se. I think it's just that the food industry is poisoning us with addictive foods and making everybody really sick. But you're right, obesity and type 2 diabetes are associated with high blood pressure. And if you look at the GLP-1 agonists, they reduce your blood pressure equivalently to traditional antihypertensive drugs. So I'm treating my patients in the hypertension clinic now with GLP-1 agonists because it treats diabetes, obesity, addictive behaviors, and blood pressure. So they're really great, yeah. I think they'll have a big effect.
Following on from that one, Adam, the fellow that you might have known once upon a time, he gave a talk at St Andrews University while I was there, oh Christ, 20 years ago, I think. Mike Horan, and very entertaining speaker. And he put an MRI up and some other analytics, relatively crude for the time, and stated that the earliest indication that they'd got was a reduced delivery of oxygen and glucose to the brain with dementia and Alzheimer's. Now, when you're talking about vascular disease, how far do you go back? And you mentioned hypertension, but there's lots of people with dementia, and my dad was one, Alzheimer's, that don't have high blood pressure, that their blood pressure is relatively normal. And you mentioned obesity. I bring in sleep apnea. There's a strong association with sleep apnea and dementia and Alzheimer's disease. There also is with diabetes. And not all people with sleep apnea are obese or have diabetes. So is there some systemic metabolic component here that is an early warning, as Mike Horan suggested? or is it as late as you suggest, even at the vascular level?
Yeah, that's a really interesting angle, because I think just from a blood pressure perspective, 85% of the population over 65 have got a blood pressure greater than 130 millimeters of mercury systolic. And it's that level, although most people would consider that to be normal, in America now, that's classified as hypertension. And it's at that point in your blood pressure, if you measure your blood pressure at home, it's above 130 systolic, you will be developing small vessel disease of the brain. That's been shown by multiple studies. I suppose from the perspective of blood flow, it's not just an overarching umbrella. I mean, and you can see by the sorts of work that, Catherine is doing as well. Blood flow is not just about delivering oxygen and nutrients, it's about waste disposal as well. And we know that all of these risk factors impair things which, nominally we call the glymphatic system. So you've just got to have a healthy cerebrovascular disease in order to maintain healthy function. And we know that cardiovascular disease clusters. So, like you say, I mean, everything has to be looked at in totality.
Hi, thank you for the lovely talks. My question regarding the vasculature in the brain is, unlike the neurons, they can divide and grow. Just wondering if there can be a way to increase it to help clear, pathologic proteins, aggregates, and also might be helpful for, different neurodegenerative disorders.
So your question is, if you improve cerebrovascular function, would you have less aggregates of proteins like tau and amyloid? I mean, I think if you look at studies of the heart or the kidneys, your answer is staring at you in the face, because all of these conditions show aggregates of, microvascularly toxic proteins which damage cardiomyocytes, nephrons, and neurons. And I think that us, by bringing the whole field forward and seeing people 5 years earlier, gives us the opportunity to really see whether some of these therapies, like tau aggregation therapies, might work at that point. Because it may be that if you implemented them at the time at which you've got irreparable cerebrovascular damage or irreparable dementia, they wouldn't work anyway. Do you not think? Get it in early?
Yeah. Uncontroversially, absolutely. And I think that the antibody trials against the amyloid proteins are showing that as well. The earlier you're going to go, the better the chances of doing something, prevention better than cure. So, yeah.
Yeah, I would just say about lecanemab and donanemab, side effects that you get from these microhemorrhage and edema, they present with stroke-like symptoms. In fact, the only person who died from the clinical trial was somebody who presented with a stroke and they had a microhemorrhage, but they got thrombolized. And the reason that we and my colleague Harry Pritchard in Manchester, we think that you get microhemorrhages because of the anti-amyloid antibodies is because the antibody therapies actually strip away the amyloid and it attaches to vascular smooth muscle cells and it degrades them. So you get these kind of small mini pseudoaneurysms that the amyloid is actually holding in place. We've shown that the amyloid kind of destroys the arteries anyway. And so if you treat them earlier, you're going to improve your waste disposal and you'll clear the amyloid.
Right, well, such a lovely and informative session. So I've got two questions. The first one regarding the vasculature in there. It's lovely to see that there is a clinic like this in Manchester now. Do you look for example, any particular mutation for those patients that is linked with SVD in particular, such as the NOTCH3 or the CADASIL1 or CADASIL2? That's my very first question. And do you think as well from the data that you are collecting at the moment, is there any particular pattern of where the microbleeds start to happen? And on that note, and second question to Will, I know that like historically is of course like Alzheimer's disease and tauopathy start at the cortical stage. Is there any data that is looking at whether there is any particular pattern of accumulation at the blood vessels and the neurovascular unit that could proceed or go hand in hand with the pattern of distribution of the disease as well? Thank you.
So your first question, I think, was about younger, more rarer kind of genetic predispositions to dementia. So I have to be really clear. This is a sporadic dementia clinic for the over 65s. Everything that I've said doesn't really correspond to the younger dementia. So if I presented with dementia, I wouldn't be seen in the clinic. Likely, I would have a condition like CADASIL or carousel. We have a specialist neuroscience centre in Salford, and they do the genotyping. And things like CADASIL present in your 30s with migraine with aura, and there's a strong family history. I think the next question's for you.
Yeah, were you talking specifically about tau therapy? And is it...
Any particular pattern of the disposition, like of the proteins around the neurovascular unit, like at the beginning of the disease?
Of the immunotherapy or the tau itself? Of the immunotherapy?
Yeah, is like any particular pattern of clearing that? Does it happen at particular... Is it everywhere? Is it, you know, any particular pattern of like that?
Right, yeah, no, it's an interesting question. So I think that the model for the antibody therapy is preventing spread. So, I mean, we don't think antibodies on their own can get into cells. They require the ability of tau to take the antibodies with them. So, therefore, it follows the tau. The idea is that it will just block the next cell. So it won't do anything to address existing pathology. So it's defined by that spread.
Hi, thanks for the great talk. I just wanted to suggest two classes of drugs that they have a lot of controversy around them because they exist within the Schedule 1 Pandora's box, but I think they have a lot of potential to treat dementia, and that's the psychedelics and the cannabinoids. So at its cellular level, they seem to both have pretty potent neuroprotective and anti-inflammatory effects. Specifically, the cannabinoids can shut down excitotoxic mechanisms that overactive and stress synapses, and psychedelics via serotonin receptors seem to promote mitochondrial survival in stress neurons. And then zooming out a bit, there's studies to say that psychedelics like LSD can promote blood flow to the brain. And then zooming out further, there's been multiple sort of studies with psilocybin-assisted psychotherapy where you see obviously changes in depression, so reductions in depression, but then also reduced rates of smoking and alcohol intake in populations that are addicted to cigarettes and or alcohol. And so I just wondered if these two classes of compounds have really been looked into much for dementia, or if there is any scope to investigate them.
I mean, I can comment from a synapse point of view. So there are, as you point out, there are a number of studies, particularly looking at the psychedelic psilocybin with things like dendritic spine growth, increasing the numbers of synapses. It's not an area we've particularly focused on. I think there are some suggestions it could be used in neurodegeneration. I don't think those, as far as I know, those the trials or anything have progressed. I guess there's a bit of care to be taken, bearing in mind the population that you're going into. But in theory, I mean, if it can grow synapses, potentially, yes, that could be used. Anybody want?
Can I talk? Just, I am very interested in the relationship between statin drugs and Alzheimer's. What is your opinion?
Statins?
Yes, statin drugs.
Sorry, was that a question about statins? Could you put your hand up? Oh, okay. So, your question was about whether statins are protective or destructive. Destructive. So the evidence is very clear. If you take long-term statins, there is a small risk of type 2 diabetes and excess of that, but it's not associated with cardiovascular disease and it's not associated with dementia. I did also just want to address one of the other points that the last person made when he talked about depression. And so if you look at the data coming out from Joanna Wardlaw's group in Edinburgh, and you look at small vessel disease of the brain, or what we would classify as vascular dementia, small vessel disease of the brain doesn't present with memory loss per se. You get very mild memory loss, but you get low mood, get apathy, loss of executive function. And when people in the clinic have it, they just talk about their patient, their parent, or their relative, kind of like the volume control being turned down to about a 3 or a 4. And so what you'll find is, so we call this kind of vascular depression, and it will be improved by treatment of all of the different risk factors, we think.
Thanks for your talk. I have a question for Professor Jackson. It's a very general question because you are working on synapses and synaptic connections. We are working on cell transplant, cone photoreceptor transplant to treat diseases like retinitis pigmentosa. I would like to know, can we use your methods to increase the number of synaptic connections between the transplanted cells and the host, in our case, host retina?
Yeah, it's a good question. So yes, in theory you could. I'm not aware of that being done, but some of the new targets coming through to increase the numbers of synapses are already shown in vitro that they increase the number in stem cells, for example. So I guess you could, once you implant the cells, then give one of these treatments or a growth factor or something which could then potentially increase the number of synapses so the cells integrate better, I guess, which is what you're aiming for.
Hello. Oh, yeah.
Thank you. Would the panel like to speculate on why the anti-shingles vaccine seems to protect against neurodegeneration and Alzheimer's disease?
Yeah, it will be speculation, but it's really interesting observation, the study from the Welsh cohort where having the vaccination reduced chances of getting dementia. As far as we know, the zoster virus itself doesn't cause Alzheimer's disease, and from what we know, we think it's unlikely to be cross-reactive protection against HSV1. So I think the main hypothesis is that it's more like the BCG vaccine, where you get a kind of altered immune system, and for whatever reason, that is more conducive to existing with protecting against Alzheimer's disease. So some sort of non-specific, non-adaptive effect of the vaccination. It's a very interesting question, though.
Hello, fantastic talk. I know you already touched on this to some extent, but what do you think will be the most promising ways to identify a dementia patient maybe 15 years before symptom onset? And to what extent would you expect these to vary depending on the type of dementia that you're looking at?
So your question is how we would best identify people at risk of dementia?
Yeah, before developing overt cognitive dysfunction.
So I think this is a really interesting area because I think that you learn lessons from other vascular illnesses. We have looked for years and years and years to try and identify people, how they might present with ischemic heart disease or chronic kidney disease or stroke, and we've never managed to do it. The best way that we have of identifying people is by looking at their cardiovascular risk factors. So if you came into a clinic and you had high blood pressure, high cholesterol, and I picked up atrial fibrillation, you'd be at risk. But ultimately, the way that we pick up patient is the patient being a biomarker themselves. So they come in and they say, oh, I don't think my brain's working properly because it's taken me twice as long to do the crossword. Or my family member is saying that my memory's not that great. Well, at the moment, they're just told to come back in 5 years' time and that there's nothing wrong with them. Or if somebody's even kind to them, they might tell them to organize their affairs. They don't even get their blood pressure checked. The patient is the biomarker. And we know from the prodromal studies that those patients who have a reduction in brain blood flow are far more likely to go on to develop dementia subsequently. And even those patients with mild cognitive impairment, 15 to 20% of them convert per year, 50% of them convert to dementia over 5 years. So from my perspective, the patient is the biomarker. And that's why we try to get CSF from them. Because trying to diagnose somebody with Alzheimer's disease, with a blood test is probably a bit like maybe somebody going to a hematologist with an anemia and them saying, well, I think you've probably got lymphoma, without taking a biopsy of where the pathology is. What we're trying to do is haul this medical specialty into the 21st century and actually make it fit for purpose and listen to patients when they actually come to present with us. Does that vaguely answer your question, at least?
Yes, it does. Because I know you also touched on using PET scans to estimate synapse density. I don't know whether that's going to be clinically translatable at some point.
So is the question whether the PET scan is translatable? Or?
Yeah, whether it's something we can actually use in clinic or whether it's just a useful research tool?
So it is. Obviously PET scans they're expensive and that makes it difficult. And so whether it will be routinely used, I think is maybe more of a practical issue. Some of the fluid biomarkers coming out for synapses, I think are going to be obviously they're a lot more easy to administer. So they would go forward. And obviously some of the data I showed, we need to be, I think, sure when we look at whatever biomarkers that we understand exactly what that is telling us. And so, is it synapse density, or could it be the signal be kind of clouded by something else?
Wonderful, thank you.
I was just interested in the comment you made about the amyloid clearance through the lymphatic system and obviously blood pressure lowering tablets. In terms of the ARIAs that have been shown in the antibody trials, amyloid antibody trials, obviously they're quite small numbers in the trials, but as this goes out to millions of people, those ARIA instances will go up. So has there been any recommendations about putting people on blood pressure lowering tablets before going on the amyloid antibodies to try and mitigate the risk of ARIAs?
No, I think there hasn't been. It's not viewed, you're getting my viewpoint of this as a vascular illness that's not universally shared. So, we're aware that as geriatricians and general physicians, we're going to be seeing these patients when they do present with these illnesses, and they're not life-threatening, I think that if you did have a condition like this and you said, you might present with a transient stroke-like symptom, but if you just managed conservatively, it will probably be all right. You'd take that chance. But I think the key is, from our perspective, we spend a long time trying to work out how amyloid attaches to small arteries and what effect it has on the signaling pathways in those small arteries. And when you put it onto a small artery that you've taken ex vivo from a mouse brain, it causes all kinds of havoc on that artery, and it sticks to it more strongly than any kind of vascular antibody we've ever seen. We just think that, if you think about it pragmatically, these patients will have had too much amyloid, amyloid 1 to beta 1 to 40, which localizes to the arteries, sloshing around their perivascular spaces for about 8 or 9 years now. So if you can get them when they first present, you're much we think less likely to incur the side effects. And also, you know, the newer drugs, the anti-amyloid antibodies, are now being twinned with like a transferrin, you know, a carrier. So I think going forward, we kind of anticipate that we'll have patients with mild cognitive impairment, and we'll be able to give them the amyloid anti-therapies based on their CSF. But at the same time, we'll be giving them to them anyway, because, I mean, if you go to a clinic in Europe or America and your blood pressure is above 130 and you're healthy, they're going to treat your blood pressure, or they're at least going to do a cognitive assessment. So it's a very interesting area, because it opens up a whole new area of research, which is, you know, what are the effects of, you know, a holistic cardiovascular risk reduction in patients. And like you said, it's very likely that if you do improve the brain blood flow, you'll reduce the side effects of these things.
So, thank you. Do either of the two of you want to finish off with like any sort of last thoughts? Because then I think we're going to have to close the session, but I want to...
So my last thought is mechanisms of toxicity. I think how we get from protein aggregates to toxicity, it really isn't clear, and work like you suggest could be direct like that. But for tau, for instance, it's really not clear why having an aggregate inside a cell is actually a toxic thing. And there are plenty of people with A-beta all over their brains who don't have Alzheimer's disease. So really understanding that variability and that mechanism, I think, is critical next.
So my closing thought really is to be aware of comorbidities, but also other conditions which increase the risk. So, for example, epilepsy, there's a bidirectional risk between epilepsy and Alzheimer's disease. And so we need to be aware of that in terms of treating people earlier. Maybe those coming with epilepsy, we should be aware of that increased Alzheimer's risk, and try and treated with anti-epileptics might actually have an effect down the line on developing Alzheimer's.
Thank you. So, are you okay? Brilliant. Well, I think, yeah, thank you again to all the speakers and you. And do you want to add anything?
Yeah, I just wanted to share a thought, and I'll talk to Adam afterwards because we are bringing this to a close, but just listening to the audience here, the capacity for open discussion, prompting new thoughts, and I've connected the psilocybin with the Cadicil model of dementia. And maybe chat to you afterwards about dilator capacity, because I think there was work on the Cadicil mouse model that suggested a reduction in myogenic tone and the possible vessel restructuring. And if you've got highly active cognitive activity going on, you're going to need to dilate the vessels to meet the demands of that area of the brain. And if the psilocybin, neuroprotective effects through mitochondria, etc., they may be overcoming some of that toxic effect of reduced oxygen supply due to vessel restructuring. Thank you to the psilocybin expert at the back.
And yeah, thank you to the speakers, and thank you all. And so let's give them all a thanks.